So our poster is diagnosing an atypical Melan-A-Positive granular cell tumor with extensive intraneural and perineural spread. Case report. Granular cell tumors, GCTs, are typically diagnosed through a combination of clinical, histological, and immunohistochemical evaluations. Typically, the defining marker of GCTs is the S100 protein, but CD68 is commonly used as well. Additional markers such as inhibin A, calretin, GFAP, etc., may also be used to aid in the diagnosis by excluding other pathologies. Granular cell tumors share many characteristics of other dermatological pathologies, including dermatofibroma, leiomyoma, and melanoma. For GCTs, positive S100 and inhibin A markers rule out other pathologies except melanoma, and so then negative melanin A and HMB45 are used to exclude melanoma, confirming GCT. When GCTs present atypically, whether through expression of other markers or histological appearances, differentiating them from other pathologies can become increasingly difficult, and clinical research for such cases is minimal. So for our case presentation, the 27-year-old male presented with two tender subcutaneous nodules on each of his hands that had been present for several months. Both nodules were approximately half a centimeter in diameter, firm, and non-fluctuant. As you can see there in figure 1. That is the one on his right hand on his palm, and that is the one that we'll be looking at primarily. So the patient had been previously diagnosed with a benign GCT on his back years ago, which had been surgically excised. After histological and immunohistochemical observation, both nodules revealed GCTs, but the nodule on his right hand was atypical, presenting similarly to melanoma. And so, I'll walk you through the graphs here. This first one, this table with the diagnostic marker, the purpose, expected results, observed results. So after confirmation of the first one as a GCT, it was presumed that the second one was also a GCT. And so usual confirmation that includes GCTs, the S100 and the inhibin A. And so there's the purpose, that was the expected results, and that was the observed results. And so it went as expected. Melanin A and HMB45 are then used to exclude melanoma because we need two exclusion criteria to confirm it. And so the expected results were negative, but our case had a positive result for the melanin A, to which we used PRAME. So PRAME has recently been identified in melanoma and has been used and has recently been shown to not be expressed in GCTs, and there hasn't been any research linking the two together for diagnosis of GCTs. And so we implemented it here to replace the melanin A. And so figure 1, that's how it presented clinically. Figure 2 shows the horizontally oriented, subscribed, elongated cells composed of spindle and polygonal cells with abundant eosinophilic granular cytoplasm and round nuclei. So it looks like melanoma. So that's why it was definitely included in the differential. There in figure 3, we see the GCT exhibiting extensive perineural and intraneural spread. It's very uncommon for GCTs, and it resembles more of a melanoma, but it doesn't really provide, it doesn't point us in the right direction. And so figure 4 is the pustule ovoid bodies of Millian, so there's eosinophilic globules surrounded by a clear halo. Again, it's just making it difficult to diagnose the GCT. And so down at the bottom, you have a positive staining for S100, inhibin A, and melanin A. And so, as we said, the problem was that it was melanin A positive, and the solution was using PRAME. And so we were able to conclude that for suspected atypical granular cell tumors that cannot be confirmed due to the inability to exclude melanoma from the differential diagnosis because of melanin A positivity, a negative PRAME staining can alternatively be used in place of the melanin A.